Williamson. She has positions both across the road at the Doherty and also, I guess, next door, almost below me, at the Royal Melbourne Hospital, where she leads the microbiology lab. And she's also got strong links to WeHi. And so in those roles, Deb bore the brunt of early struggles to detect and contain SARS-CoV-2 infection, especially in the Royal Melbourne Hospital during the early to middle part of last year. I guess that's something that she's going to be telling us about today, but I think it's going to be a more broadly based presentation than that. So with that, I will mute myself and you get started.
Thanks, Rory. And actually, I'll just preface this talk now by saying that this is largely a COVID-free talk. And I think, you know, that's all good for our mental health. So thanks for asking me to talk today. And what I want to talk a little bit about is how we've used whole genome sequencing in both clinical and public health microbiology. So I guess not so much about the technicalities of sequencing, but more about the implementation. And so I'll just provide a very brief overview of what sequencing is about, provide some applications in hospital setting and public health, and then think about some future directions. So look, many of you will not know what we do in the clinical micro lab, and it's I guess a bit of a black hole even to many people in the hospital. And essentially we provide information on what's growing or what pathogens are present in clinical samples from patients in the hospital. And my lab at the Royal Melbourne Micro Lab provides services to Royal Melbourne and to Peter Mac as well. So essentially, our traditional workflow at the moment is that we take a patient sample. Usually we culture that on agar plates or sometimes in nutrient broth to try and detect the presence of a pathogen, and that specimens obviously may be polymicrobial as well. We identify them, and the gold standard at the moment for rapid pathogen identification is something called a MALDI-TOF, that's mass spectrometry, and that's really been a disruptive technology in the clinical microlab. We can identify pathogens, you know, based on their spectral profiles within minutes, very, very cheaply for, you know, less than $1 per pathogen. And then we undertake susceptibility testing on clinically significant isolates, so where we know that antibiotics are likely to be used for treatment. We do that using the old-fashioned disk diffusion method or e-tests, but more recently, over the past, well, I say recently, but in generational terms, you know, over the past 15 to 20 years, there's been high throughput automated susceptibility testing machines. If we think that toxin presence or absence is relevant to a clinical infection, for example, a Clostridium difficile infection, we might detect the toxin. And if we think there might be an outbreak in the hospital, then we can perform strain typing on it. So you can see that that's, you know, a pretty iterative process. And from sample to susceptibility testing can take usually a median time of about 24 to 36 hours, but sometimes it can take a bit longer than that. And, you know, really, we want to know whether we can do better than that. Over the past 5 years, largely in public health microbiology, so not so much in the clinical lab, we've had whole genome sequencing, again, another disruptive technology. What does that do? Well, it essentially means that we can consolidate all of those other tests into one test, so if you like, a one-stop shop. That's a compelling narrative, and it's not quite there yet, and I'll talk about why that is later on. But essentially, if you have the genome sequence of a pathogen, then you can derive a lot of that other information, and we call it deriving it in silico. So, you know, on the bench, you can take a lot of that information from the genetic code of the organism. So we can identify it, we can detect antimicrobial resistance determinants, we can look for toxin genes, and we can do strain typing, you know, based on the whole genome sequence of a pathogen. So there are a variety of different sequencing approaches, and this is I presented this to epidemiologists quite recently, and I guess wanted to keep it as simple and as conceptual as possible. And this is a slide that's often used in these talks to provide examples conceptually of what the differences across different sequencing approaches are. So, we can conduct sequencing on genes of interest. So one gene, amplicon sequencing, the whole genome sequencing, but we can also do it on collections of genomes, and that's called metagenomics. And we might use that, for example, if you take a clinical sample without the culture step, and I'll come on to that shortly, and you sequence everything, and you may have more than one pathogen there. Broadly speaking, whole genome sequencing can be described as the study of the entire content of genetic material. So, bacteria, for example, may or may not have plasmids. And when we do whole genome sequencing, we're not just looking at the chromosome, we're looking at all of those extra chromosomal elements as well. And that's really important because often those extra chromosomal elements, particularly plasmids, are incredibly useful when we're looking at things like drivers of antimicrobial resistance or virulence genes that are spreading across different organisms. And so those extra chromosomal elements and mobile genetic elements, they're a critical part of whole genome sequencing. And essentially we take, looking at one organism alone, I guess is useful, but only in very distinct contexts. It's really when you look at collections of pathogens and you compare the genetic sequence, and then you use mathematical models to infer a phylogenetic tree, and you look at the relatedness of pathogens to each other. That's where the power of this technology comes in. I just want to back up and talk about some important nomenclature here. So far I've largely talked about culture-dependent sequencing, which is when you culture and then you sequence. But I alluded to metagenomics, which is what we call culture-independent sequencing. So when you sequence directly from patient samples. And you'll have heard things like short-read sequencing, which is largely still the Illumina technology, versus long-read sequencing. And that's Oxford Nanopore and PacBio are the leaders there. And each of them have pros and cons. So let's think about culture-dependent sequencing. So this is, again, where I mentioned we take a patient sample, we culture it, we get a purity plate of the organism, so we get the organism pure in culture on the plate. We extract DNA, we do some quality control steps, we do our library preparation, and then we sequence. So that's a culture-dependent whole genome sequencing approach, and that's the vast majority of whole genome sequencing in microbiology labs is this culture-dependent approach. So still takes that step of anywhere up to 24, 36 hours to get the culture before you can do the sequencing. The converse to that is culture-independent sequencing, where we take the clinical sample, we extract the DNA directly, do the library prep, and we sequence there. So it misses out that culture step, and that's really where we can start to get gains in time efficiency. So that's the first part of sequencing. The second part is obviously the bioinformatic analysis. This is with Illumina. We get these sequencing reads, which are short. With Nanopore, the reads are longer. And then when we get these reads, there are two approaches that we use in the microbiology lab. One is mapping of the reads to a reference. And the other is something we call de novo assembly, where we scaffold and we assemble the reads together to get very long nucleotide sequences. That's when we can start to look for things like resistance genes or virulence genes. And then collectively, when we have that information, these are the things that we can do, and then we report the results out. All right, so so far what I hope that I've done is given you an overview of what we do in the clinical microlab and public health microlab, where sequencing may play a role and how it might fit into workflows. Now I want to think about how we apply that. So where can we actually apply that technology to gain knowledge and use in the laboratory? So if you think about it, in a lab, when something grows on a plate and we identify it, that's a form of strain typing. So simply by giving an organism a name, whether it's Staph aureus or E. coli, that's a form of typing, because immediately we're classifying it into a distinct group. When we look at an antibiotic resistance profile, so say for example, we say the Staph aureus is an MRSA, that's more strain typing in a way, because we're saying, okay, it's a Staph aureus, but it's also an MRSA, so we're still classifying. We would call that low resolution typing. And that's really what we do in the hospital lab. Beyond that, we can do things like multi-locus sequence typing using multiplex PCR. And that is medium resolution strain typing. So it can lump things together, but beyond that, there's still more resolution that you can gain. And whole genome sequencing is sort of at the tip of this, really, in terms of resolution. So it provides almost like a forensic level of resolution based on what we call SNP, single nucleotide polymorphisms, between pathogen sequences. And really, when you get down to those SNP differences, the resolution is quite, it's several orders of magnitude beyond multi-locus sequence typing. And then by looking at those SNP differences between pathogens, that's when we can start to define transmission networks. And I'll give some examples of that shortly. So we can define transmission networks in a hospital, for example. We can define transmission networks in a foodborne disease outbreak. We can define transmission networks when there's a breach in hotel quarantine from a certain virus. And, we can look at things like STI transmission networks. So I've essentially gone through this, but when things are identical, then we say that is closely related. We never say definitively that there's definitely transmission. We usually say possible transmission. But obviously, if things are aligned and there are a lot of differences, then we say they're not related and transmission is unlikely. We also use the core SNP data. This is a slide from Torsten Seaman, who's a bioinformatician at the Doherty. And we use the core SNP data to produce a phylogenetic tree. You may have come across this slide in other talks. It's been used extensively now, but I still think it's one of the best ways of showing differences between pathogens. So, for example, a peppermint crisp and a mint patty, quite closely related there. But the banana here is the distinct outlier amongst all these chocolate bars. When you're looking at phylogenetic trees, it's important to note that the vertical distance here is largely aesthetic. So it's really so that you can see the topology of the tree. What's relevant in a phylogenetic tree is this horizontal distance. So the sum of these horizontal distances, and there should be a scale. This is terrible. There's no scale on my phylogenetic tree here. But the sum of the length of the horizontal distances between the two branches indicate the genetic distance between the tips of the tree. And that's important, and I'll talk to you about that shortly. All right, so what are some of the applications of whole genome sequencing? Well, here is, for example, how we've used it to look at hospital-level transmission of VRE. Now, the first take-home point here is that the genomic data in and of itself, when you're applying it to clinical and public health microbiology, is not that helpful. What is helpful is when you integrate the genomic data with epidemiological data. And sometimes we call that metadata. You might see it in papers where they talk about metadata. And that metadata can take many forms. It can take, for example, it's descriptive data that put the genomic data into context. And here, for example, you can see that the metadata consists of hospital A, B, C, consists of different ward names, consists of whether the sample type is a clinical sample or it's a screening sample. And basically what you can see here is that there's a cluster here with these patients 1 to 9. It extends largely in hospital network B, and it's largely from screening samples. And that's just an example of how we integrate genomics in the epi. This paper is probably one of the first in the fields to do that, and it was looking at prolonged outbreak of MRSA in a special care baby unit. They used the genomic data to look at whether or not there was an outbreak in the special care baby unit, but they also used it to identify reincursions of MRSA into the unit after the unit had been cleaned. And they were also able to contextualize their genomic data and identify isolates in the community which were linked to the outbreak via household transmission. And as I say, this is probably, I mean, I hate to use the term landmark, but it was a landmark paper in the field. We can also use genomic and epi data to link up not just transmission between people, but link up transmission between people in the environment in the hospital. And this is an example of how genomic data were used to link up contamination of water supplies with patient infections. But again, the take home here is that you can see that there are different ways of looking at genomic data, and there are different ways of linking it up with the metadata. All right, so that's how we've used it in hospital settings. I want to now talk about how we've used it in a zoomed out, if you like, public health context. And I'll give you an example of some work that we've done with Shigella. Now, Shigella is a major cause of diarrhea globally. It's a cause of bacterial dysentery. And there are two main species of Shigella, Shigella sonnei and flexneri, and it has a very, very low infectious dose. It's one of the hallmarks of Shigella infection. So as few as 10 bacteria can make you really sick. And when people need antibiotics, these antibiotics are generally the ones that we would give, so ciprofloxacin or azithromycin, and in very severe cases, ceftriaxone. Now, the incidence of Shigella has really skyrocketed over the past 10 years in Australia. Obviously went down in 2020 for a number of reasons, social distancing, impaired access to testing and treatment, for example. But anyway, the take home is that Shigella has gone up a lot over the past 10 years in Australia and actually in other developed countries. We were interested in why it had gone up. I mean, if you look at this slide, I'll just go back. This slide in and of itself isn't that helpful. I mean, it tells you that there's a problem, but it doesn't tell you what's driving it. So what we wanted to do was to integrate the genomic data and the epi data to try and work out what was driving that increase in incidence in Shigella. And we undertook sequencing on just over 500 Shigella isolates from Victoria, and we integrated it with epidemiological data on sexual risk, and I'll come onto why that's important, and travel. And this is what we found for Shigella sonnei, so the main species of Shigella. So remember, the take home was that the horizontal distances are more informative in terms of distance between isolates. So you can see here, there's when you add up this and this, there's quite a lot of genetic distance between this outlier group and these ones. But here you can see this one here, there's very little genetic distance between the isolates, and that's indicative of an outbreak. And essentially there were four main lineages of Shigella sonnei. So 1, 2, 3, and 4. So in and of itself, remember I said that's not particularly useful. However, when we overlaid it with the epidemiological data, what we found was that these lineages, 1, 2, and 3, were largely associated with travel to distinct parts of the world. This lineage here was associated with travel to the Southwest Pacific, here associated with travel to Southern and Central Asia, and here Southeast Asia. But here, this one here was associated largely with the gay male community, men who have sex with men. And we'd had sort of an inkling of that from the epidemiological data alone, but this really confirmed our suspicions, was that there was a large outbreak of Shigella in that community in Victoria. When we overlaid the antimicrobial resistance data, what we found was that ciprofloxacin resistance was largely associated with travel to Southern and Central Asia, but azithromycin resistance was really mostly found in the MSM-associated lineage. And we looked into that further. And what we found was that when we looked at the plasmids in this lineage here, azithromycin resistance was associated with a gene called MPHA on this very resistant plasmid, that was really hadn't really been described before. A group in the UK were also working on this at the same time, and the beauty of genomic data is that it's universal and transferable. So we were able to share data with them, and essentially we found that this was a global plasmid outbreak, and this plasmid, multi-resistant plasmid, could cross species. So we found it in Shigella flexneri as well. And if you think, if we just looked at that graph alone, looking at the increasing incidence of Shigella, you wouldn't have any of this information. But as I said, the power of genomics and integrating that with the epidata really gives you these vast insights into what's going on. And this is really a perfect storm, this Shigella story. So we've got these very dense transmission networks. We've got a pathogen with a low infectious dose and a highly resistant plasmid, which can cross species. And we've got why is it azithromycin resistant? Well, this particular demographic group actually is very exposed to azithromycin, because azithromycin is used for the empiric treatment of sexually transmitted infections. So we hypothesized that this was really a selection pressure here, again, feeding into this perfect storm. The story doesn't stop here, actually. We actually saw an increase in this lineage here, and this lineage here, which is the ciprofloxacin resistant lineage, really took off this year, and so this one has become the dominant one here, so resistant to ciprofloxacin, and it acquired that azithromycin resistant plasmid, essentially meaning that Shigella sonnei is a almost untreatable pathogen with oral antibiotics in our setting. All right, and I'm just going to finish with a brief example here of another use of genomics, and this is really at a global level, and we were involved in this. And this is a global outbreak of a non-tuberculous mycobacterium. So a few years ago, there were some initial case reports of patients who had cardiac surgery and then picked up an infection with a non-tuberculous mycobacterial species called Mycobacterium chimera. And these infections started popping up in different countries, and it's rare. This was almost like a previously unheard of pathogen, and immediately that raises epidemiological red flags when you've got this. This is all probably sounding a bit familiar in the current context, but you have a never seen before pathogen popping up in multiple countries, suggests that there is a global outbreak. But in this instance, all infected patients had undergone cardiac bypass involving, we call them heater-cooler units produced by a single manufacturer. Again, red flags about a point source outbreak here. And this was investigated epidemiologically, but there was nothing to bring it together. There was no definitive proof that these infections were linked. What are heater-cooler units? Well, they're used in cardiac surgery to essentially heat or cool the body temperature depending on what part of the depending on the stage of surgery. And this is what they used to look like. And these are in the operating theatre, and there are water tanks in here. The patient's blood never comes into contact with the water, obviously, in the tanks, but the water tanks are used for heating and cooling, almost a bit like an evaporator. And the water evaporates and is vaporized out of the front of this machine. Intuitively, you would think that that's not a great idea in a sterile theatre, but that's what used to happen. And so you can see that if there was contamination of these tanks and it was blowing out into a sterile field, then this could seed an infection in a patient. So these infections occurred in Australia, across different states, and there was some in New Zealand as well. And we isolated them from 43 were from heater-cooler units, and we also had some patient samples as well. And so we performed whole genome sequencing on these Mycobacterium chimera isolates, and what we found was that yes, there was this clade of Mycobacterium chimera, and we had samples from Perth, we had samples from Brisbane, we had samples from Sydney, from Auckland in New Zealand, and they were all essentially in the same clade. And here's what really clinched it. So this little subclade here, this one here was a patient infection, and these two are from the heater-cooler unit, the same heater-cooler unit that was used in that patient's cardiac surgery. Again, you know, if you're working on something topical, then chances are that somebody else is working on it too. And this group, led by Kieran Perkins at the CDC in Atlanta, were also working on it, and they contacted us about sharing that genomic data. And we did, and they had an outbreak at a hospital in Pittsburgh. And so we shared our genomic data, and sure enough, the red stars are the isolates from the US. You can see that they are present in this subclade here. And less than 10 SNPs, now in something that has a genome of 7 million base pairs, when you've got less than 10, or in this instance, you know, no or 1 or 2 SNPs separating out those bugs, that's highly suggestive of a point source outbreak. Okay, so what have we talked about then? So what I hope that I've shown you are some of the applications of genomics, applications in the hospital setting to look for hospital outbreaks, applications in a public health setting to look for local outbreaks here, and also on a global scale as well. So, you know, this is where we used to be in microbiology. It was, you know, we used to have this kind of onerous workflow for characterization of outbreaks. We're now here, I think, where we've really replaced a lot of these with whole genome sequencing, but we want to get to here, mainly in the clinical microlab, whereby we can take patient samples and sequence them directly and make a rapid identification of what's in the samples. But, you know, it all sounds very, again, conceptually attractive, but there are some challenges for implementation. So the turnaround times are, it's hard to beat that MALDI-TOF for identification, and we need to get those turnaround times shorter with sequencing to, you know, to be used in routine care. The costs are high, but rapidly decreasing. And actually, when you do cost-benefit analysis, you know, this technology could potentially replace multiple tests. Bioinformatics, actually, I would say used to be more of a bottleneck, but it's now kind of becoming a bit democratized in as much as there are now much more friendly, user-friendly bioinformatic tools available. Reporting of the data is an issue. So, you know, people are used to having test results reported to them in a certain way that hasn't changed in many, many years. So how do we report on some of these genomic results? That's a large piece of work. And I would say that if anybody is interested, please feel free to contact me. Very happy for people to come and have a look around the lab, COVID permitting, and happy to talk to anybody further who wants to get involved in genomic projects or just learn a little bit more about it. There are heaps and heaps of training courses online about this, and lots of very user-friendly conferences that are always up and always available for genomics, public health genomics. All right, and thanks very much for listening. And Rory, happy to take questions.
Thank you. Just switching myself back on. Thank you. That was really great, really informative, even for people like me who should know all of this stuff already somehow. So I'm not sure if anybody currently has their hand up, but we did have a slightly off-beam question in the chat, which I'm happy to read out, or maybe Max would like to read out. Are you there, Max? So maybe that was from, I think that's right. Other people can see this chat question. It says, perhaps unrelated, but to your knowledge, how good are whole genome technologies at capturing the full microbial diversity? For example, gut microbiome species diversity. If it can't, is it just a limitation of the system/technology? Maybe unfair to go straight in with that one, but then if you have anything to say on that subject. I've got two of you now.
Sorry, Rory, I think I dropped out there.
What? Yes, my screen now has two of you. One of them's frozen and the other one is moving. Shall I just try again? Perhaps unrelated, but to your knowledge, how good are whole genome technologies at capturing the full microbial diversity? For example, gut microbiome species diversity. If it can't, is it just a limitation of the system or technology?
Yeah, that's a good question, and I get, yes, it's there are certainly others who would know a lot more about this than me, but it's much better than the current methods we have, which is culture, basically, because a lot of the gut bugs would be anaerobic and require very specialist culture systems. The limitations are certainly on the technology and the depth of sequencing. And so, for example, there's a trade-off at the moment between what we do, which is, putting more sequences on a run and reducing the depth. But if you're willing to blitz it and throw everything that you've got with one sequencing run, then you can sequence pretty deep. And in fact, Rory's a specialist here. He's done a lot of work on deep sequencing of clinical pathogens. So you might want to comment.
I think my comment was really just relating it back to one of your kind of examples, which is the VRE, the vancomycin-resistant enterococcus. And that is, sure, you could use sequencing to try and find VRE in screening samples, but you'd have to sequence very deeply to find something which might only be there in a very small proportion of the total bacterial mass, and yet be very, very important. So you'd sequence a lot of stuff that might not be very interesting before you found the thing you're interested in. So that would be my kind of, yeah. And then also methods to put back together genomes are getting better and better, especially with longer reads. So again, the problem though is that some of the interesting stuff might just be 1/1,000,000th of the total mass. And so there's a huge amount of sequencing. What you want to do is put it all back together. I've got some more questions. It's always, so the top of my list is William Vo. You'd like to unmute yourself and ask the question?
Hello, can you hear me?
Yep.
Thank you so much for the talk, Deborah. I guess my question is on costs. I heard whole genome sequencing described as quite expensive. What is the reason for that? Is it the reagents? Is it the technology needed? Or is it the training?
Yeah, that's a great question, and it's a composite of all of those, actually. So it's the labor costs involved. Actually, I'll back up a bit. It depends on where you want to do your whole genome sequencing, and it depends on what's on offer. So I would always check that first. So, for example, it depends whether you incorporate the bioinformatic analysis into the cost, whether you do the DNA extraction and library prep yourself. But the cost is essentially a composite of reagents, labor, overheads or for the facility that you're using. And that they would be the main drivers of the cost, plus or minus bioinformatic analysis. Honestly, whether or not they multiplex, the depth of the sequencing, there's a whole range of things that contribute to the cost. But honestly, the cost has come down, enormously over the past 10 years or so. And really, in this day and age, we shouldn't be charged more than, $100 to $130 for sequencing per pathogen. Does that answer your question?
It does. Yeah, thank you.
Yep.
So Harry Dempsey is the next person.
All right. Thank you, Deborah, for your talk. What I wanted to ask is, how long does it take? I remember you said that it's a big bottleneck, the time required to do whole genome sequencing of, of a pathogen a patient might have. How long does it take, and what is a reasonable target length of time for trying to get to so that we can help patients relatively quickly?
Yeah, that's another great question, and there's no one size fits all, Harry. So, for example, if you thought that a patient had if it was a pathogen that required culturing before you did whole genome sequencing, then because you were looking at strain typing, for example, then it might take 12 to 24 hours for that pathogen to grow on an agar plate. It might take at a minimum another 24 hours to get the sequencing through, and that's at a minimum. So you'd be looking at a couple of days really minimum. But for things like SARS-CoV-2, for example, that's sequencing directly from clinical samples. Obviously, we don't culture that before we sequence it. And so some labs have actually got that down to, anywhere sort of 8 to 12 hours, sequencing directly from clinical samples. So it's an amplicon-based approach where there's multiple amplicons and then subsequent sequencing. And also it's obviously a much smaller genome than a bacteria. It's 30,000 base pairs as opposed to millions of base pairs. So no one size fits all. It depends on the question that's being asked as well. What metagenomics can do, and this is actually a large project that we're working on at the moment, is trying to evaluate the use of metagenomics for clinical diagnosis. But I would say that for some patients, for example, with life-threatening bloodstream infection, you need a diagnosis in hours, not days. And when I say hours, I mean like a few hours, and we're not there yet. So unfortunately, there's no short answer to your question. It depends on the host, it depends on the question being asked, it depends on the pathogen and the resources and facilities available. Does that answer?
Yes, that does.
Yeah.
Yeah, especially I think mostly I imagine it mostly depends on the pathogen. If it's something where you can have it for a while and it doesn't make much of a difference, if you're infected, it's okay. But if it's, if you need to be treated within hours, then it's really, really important.
Yeah, that's right.
Yeah.
And I mean, I love genomics, but I'm not naive enough to think that it's going to replace entirely everything that we do. It's complementary, very complementary to a lot of the things we do in the lab, but it may not replace some of those other things that we use for diagnosis.
Yeah, cool. Thank you.
Can I just ask a sort of a kind of a follow-up version of that question? So how do you make sure that, the question is sort of like, you're not every day in the lab trying to fill up a whole large sequencer full of genomes, typically, right? So how do you make sure that you can always have enough capacity available, like, so that the next urgent thing coming through can sort of simply slot into a run that you were going to do anyway? Essentially, so that when it's very urgent, it doesn't just cost thousands of dollars each time because you have to drop everything. So it sort of fits into a nice routine kind of workflow. Yeah,
you ring fence space.
Okay, so somehow you're doing stuff on a routine basis, and then you can pop in a few urgent things which then get done on the run you were going to do anyway, and so you're not spending all the money for that, potentially thousands of dollars for that run too.
That's right. You ring fence space and you have a diversity of testing platforms. So you might keep a MySeq for, or NovaSeq or whatever for something urgent. And you put the, other things on a NextSeq or even a HiSeq, depending on what you've got.
Yes, we're not going to go into all the different sequences and capacities, but there are small ones and big ones.
Yeah.
For the purposes of this conversation, anybody who wants to find out about all of that, they can come to either of us, I guess. There was somebody who sneakily had their hand up and may have taken it down again. How are we looking? So I had a kind of another trying to sort of stimulate discussion kind of question. So just, and bear with me. So what is it that stops you from using a mass spectrometer directly to identify things in clinical samples? What is the kind of intrinsic problem with that as an approach? Yeah, so not much of the stuff you want to detect, even if there's a peak that absolutely pins down that organism.
Correct. Yep. Background noise. A lot of diversity of background noise across different clinical samples and different patients.
Okay, so now this is the really stretching question: is does that help us to understand how we could best apply next generation sequencing to uncultured samples? Are we looking for particular kind of signatures that will tell us if we've got something? Is that the way to think about it?
I think the knowledge there is in this is where the domain knowledge comes in interpreting junk from things that might be real. Yep. So for example, if I were to sequence a skin swab, right, and I found that there was a group A strep in the high abundance in the skin swab, but there was some staph epidermidis and some, commensal skin flora also in the skin swab, I might say, well, that's probably, and using my knowledge of clinical microbiology and what causes infections, I would say that group A strep's important, but the other stuff is probably just crud. Yep. And we do that in culture anyway. We might grow junk on a plate, but there'll be something there that's important, and the same applies to whole genome sequencing. Yep. Right. But at the moment, the depth that you get from them, there's too much noise to ever get that from mass spectrometry at the moment. Anyway, based on the ones that we use commercially, I don't know. Andrew Webb might have some much fancier machines, but yeah,
they'll still be expensive to buy, and you'll have to
correct
time on them and prepare the samples correctly and all that, I guess. Yeah, no, I was just, I guess, thinking about what the signal was that you identified, because you sort of slightly glossed over it, like epidermidis, we know shares quite a lot of sequences with other potentially pathogenic or more likely to be pathogenic organisms. So a lot of that you'll sequence and you have to sort of throw it away because it's not informative. Is that? Okay.
Yeah.
Okay, we do have somebody else. Orally, are you there? Can you unmute yourself?
Yeah, thank you for such a clear presentation, Deborah. I was just wondering, are there any strategies put in place to reduce the spread of the plasmid PKS or 100? I think that's the name.
Oh, that's a great question. Yeah. Are there strategies in place? Yes. Not specifically aimed at the plasmid, but much more at a public health level. So, for example, an educational messaging or health promotion, I should say, messaging around, things like antibiotic prescribing for gastroenteritis, or making sure that if you've got gastroenteritis to go to your GP. The GP has to culture the specimen so we know what's there and we can do susceptibility testing. But also around things like, not having, abstaining from sex, for example, if you're infectious. So there's a whole bunch of things that go on which are ultimately aimed at stopping the spread of the plasmid, but they're not specific, plasmid interference strategy, things like that. What we do know, interestingly, from in vitro work is that plasmid's not particularly stable. It will drop out very easily in laboratory passage. So intuitively, if you scale that up to a population level, then you might think, well, if we can practice better antimicrobial stewardship and not have as much antibiotics being thrown around, then the plasmid might it might not have that selection pressure and that really attractive environment in which to spread. Does that make sense?
Yeah, it does. Thank you very much.
Yeah, I quite like that answer because we sort of think of the azithromycin resistance as being kind of deterministic. If it's there, we're eventually going to see it and we'll just have a disaster. But I guess there is a model you can run to say, well, we want to reduce the chance that it will get through the population. And I mean, do you kind of ever think about potential weaknesses? So like, why do you do stewardship? Because you rely on a particular drug, but you can see something coming on the horizon, which is just going to make the drug less useful for other things, let alone the thing that, like in this particular case, it was quite striking that suddenly the drug of choice went to being not useful. Okay, I think, oh no, Harry's hand is up, but that could be from before. Are you wanting to ask another question, Harry?
Oh, yes. Jose can go first, though, because I think his one is related to mine, is in the chat.
Oh, okay. I haven't been monitoring the chat. Jose, are you able to switch on to read your question, or do I have to find it in the chat? Here we are. Oh, I've got it. I've got it. For urgent samples, is the workflow predetermined for microbiology samples, like COVID sequencing, or depending on how bioinformaticians analyze the data? I mean, yeah, that's an interesting question. And by the way, how fast is the current standard strain typing in hospitals?
All right, is the workflow predetermined? I'm not quite sure what that means, but I mean, by their nature, you don't always know when urgent samples are coming, so you have to be prepared at all times. But yes, you know, in laboratory practice, workflows are largely predetermined and standard, and the reason for that is, you know, we are reporting out on things that may change the way a patient is treated or may change the way that public health decisions are made. And so those workflows have to be standard. They have to be standard, and we aim to get them accredited by regulatory authorities. And so standardization, it might sound boring, but it's hugely important in the laboratory. You have to do the same thing every time, because if you don't, you're introducing variability into it, and it makes your results less reliable. It's like doing a lab experiment, right? So you have an SOP and you follow it, and you need to be able to reproduce it. And the same applies, and at a much more stringent level in the laboratory. So the sequences are generated in exactly the same way. We run controls through that, and the data are analysed by bioinformaticians. You know, obviously in a research context, there's much more freedom to move, and you know, you can do much more exploratory data analysis. But in the laboratory setting, when we're reporting out on things that might change what people do, then the data are analysed in the same way. How fast is the current strain typing in hospitals? Again, it comes back to that other question, which depends on the pathogen, depends on the question being asked, depends on the technology.
So then, just widening it out, if you have a particular kind of sample, you have a protocol which sort of says, if I find this, then I move down this path, and then it can switch. It's not, so maybe the question was partly about, like with COVID, it's a very simple if then, if this, then that. If you don't get a positive PCR result, then you don't sequence, for example. But if you do, then you try and sequence, and then you kind of know what to expect in terms of whether you get a sequence, but it's very standardized and kind of simplified. Okay, so I think Aurelie, you may have another question, or she might.
Do you mind if I ask us to follow up to that one?
Sure.
Thank you. So I guess related to that question, I remember you said that the bioinformatic analysis isn't necessarily such a bottleneck. But can you explain to me again the challenges for the bioinformatic analysis in order to tell a patient what kind of pathogen they have quickly? Because I imagine it would be a lot easier to prove the bioinformatic analysis by just having more computing power, like compared to trying to develop completely new methods in order to get the whole genome sequencing to less than a few hours, for example.
Yeah. Okay, there's a lot to unpack there actually, but so it's not so much a case of the technology. I alluded to this, and I didn't go into it too much, but it's I mentioned the reporting is a challenge, right? And it comes back to Jose's question as well around standardization and quality controls and quality assurance. So when we report stuff back that might change what happens to a patient, bearing in mind that when you change patient management in a hospital, there's a whole framework of clinical governance around that and liability. And, you know, if a patient is given the wrong antibiotics because of crap information that comes out of the lab, then you're liable for that. The organization is liable for that. And that means that the workflows need to be totally standardized, totally accredited. So at the moment, and this is actually the project that is ongoing at the moment, is looking at locking down workflows, both sequencing and bioinformatic analysis, so that we can standardize those to report results that can be used for patient care. So say, for example, my research lab, who are looking at metagenomics, were to use one bioinformatic analysis or one bioinformatic approach, and they found that a patient had an E. coli in their blood, one day, but they thought, oh, you know what, I might just try this new program that somebody's published, and they found that that patient had a Staph aureus the following day. That's an extreme hypothetical example, but if I had said, oh, thanks for that information, I'm going to give that patient antibiotics to treat their E. coli, but it actually turned out to be a Staph aureus, and they didn't get treatment and they died. Again, it's extreme hypothetical, but you just can't do that. You need lockdown protocols, and so that's what we're working on. Is that sort of what you were asking, Harry?
Yeah, exactly. So I guess the bioinformatic challenges are really to make sure everyone's using a standardized protocol, and yeah.
That's exactly right. So it's the programs, the pipelines, they exist for looking at metagenomic data. In fact, there's lots of them, which is a challenge in and of itself, because there are a lot of them. Which one do you use? Which one's the most accurate? Which one's the most reliable? Which one's the most user-friendly? So there's a whole bunch of things. So the programs are there. It's a case of distilling them down into a standardized workflow.
Yeah, cool. Thank you.
Okay. So I can't see any more questions either in the chat or people's hands up. So I guess it's a chance to just wave or whatever and say thank you to Deb for a really stimulating conversation and some really interesting questions and answers. Thank you very much.
Oh, thanks, Rory. Thanks, everyone, and thanks for the great questions. I'm happy for people to contact me offline. I think Rory's got my details. Thanks so much.
Yes.
Cheers.
Okay, thank you.
Thank you.